Right, good morning, everyone. Thanks a lot to Tom, in particular, and to all the organizers for putting together this amazing meeting. I've been learning tremendous amounts of new information, and just really a lot of fun to be here. And also thanks a lot to the previous speakers, to Monica, Kevin, and Jella, for making my work extremely easy, because they have introduced all the concepts I'm going to be operating with. So basically, I can just run through the first 20 slides and then give you the results afterwards. But first things first, so my title is Temporal Hologenomics, and that's actually a term that for the first time I heard from Tom, because I was kind of reducing it to metagenomics. It's like, no, it's more than that. And if you have difficulties understanding what it is, AI does too. So in contrast to Kevin, I asked AI to generate a picture of temporal hologenomics. That's what it came up with. It's not completely clear to me, but maybe it will become clearer as we go on. And I think the way I'm thinking about us and about biology in general is that change is ubiquitous. It's a definition of life. Life is change. But this change has been dramatically accelerating in the last few decades, last few hundred years, and primarily because of human activities. So if we think of climate change, habitat destruction, conversion for agricultural and urban developments, pollutions due to our industrial production and biodiversity loss. And of course, all of that led to this proclamation of a new geological time period. Initially, it was suggested to be a new geological epoch. It was downscaled to a time period. But you've seen all that, so I'm just going to quickly highlight a few aspects that I think are really important. One of the things that defines Anthropocene are these technofossils. Plastics, concrete, black carbon, but also human-produced fertilizers, pesticides, and antibiotics that get out into the environment. And if we think of the microbiome or the holobiont, this will have a huge effect on these interactions. The other aspect that I would like to highlight is biological change. And when I think about biological change, I think about it in two different categories. Extinctions, shown here with an accelerated rate of extinction in mammals, for instance, in the last 250 years, compared to the background that spans almost the last 2 million years. And population declines, so that on average, vertebrate populations have declined by 70% since the 1970s. So from this perspective, I think we have, as biologists in general, we have been considering these factors and how they affect the macroscopic organisms. So a classical example of industrial melanism, how human-produced contaminants have affected biology and ecology of the species, and a work that Tom van der Valk, who's here in the audience, carried out when he was a PhD student, showed how population declines affect the host genomes, specifically by comparing museum specimens that predate the most recent declines in gorillas. He was able to show that historical populations had significantly higher genetic diversity than present-day populations, and that inbreeding has dramatically increased in present-day populations compared to historical populations. So he used this temporal approach to look at the genomic consequences of decline. But what we frequently miss from this picture are the microbiomes. So what about them? And I think a kind of conceptual image that I would like to show here is that host-associated microbiomes, which thankfully I don't have to introduce to this audience, are primarily influenced by two factors: by the external environments, the temperature, salinity, pH, and the internal host environments, such as host genetic factors and particularly immune factors. So these two things are the ones that are dramatically changing today, right? So we have climate change, habitat change, pollution on the external side, and we have population declines on the internal side. So we're changing both the environment and the host genetics. And of course, with this, we're changing the microbiomes, and the microbiomes are absolutely essential for these fundamental host biological processes, anywhere from digestion and immunity to reproduction, behavior, and adaptation. So I would argue that if we want to understand how these changes are influencing the host microbiome interactions, we need to take a temporal approach. And for that, we need time travel. And ever since I moved to the UK, that's the only acceptable image of a time machine that I'm allowed to use. So we need temporal sampling from the time periods that predate and span the change to be able to compare to the present-day situation. So in terms of human changes, we're talking about the last, 150, 200 years? Of course, there's a challenge, and that is that microbiomes usually do not preserve over these time periods. If you think of the skin microbiome or the gut microbiome, this is going to degrade as the host dies. But as you have seen in Jela's talk, there is a rescue. So there are these microbial fossils, and I will be talking about our favorite microbial fossil, the dental calculus, so the preserved oral microbiome. Why should we care about the oral microbiome? I would argue that the oral microbiome is actually a uniquely suitable substrate, if you like, to look particularly at these human-induced changes. So it sits at the intersection of the internal and the external environments, which means that they're going to reflect both sides of this equation. It is connected to the respiratory and digestive tract, and with that, it not only reflects what happens in the mouth, but also beyond, as Jela has shown in her presentation, reflecting, for instance, the changes or the stability of the rumen microbiome. It is a driver of oral and systemic diseases. Oral diseases we can think about caries, periodontitis, but systemic diseases involving cardiovascular diseases, diabetes, cancer. And it is a potential contributor to local adaptation, for which I will show you a story later. So this sounds great, but how abundant and how ubiquitous is dental calculus? And a PhD student in my group right now in Edinburgh, Johnny Richards, together with his partner in crime, Markella Moraito, who I'll introduce in a sec, went out and scored hundreds of specimens, thousands of specimens of more than 140 mammalian species in European museums, trying to see where dental calculus is present. So what you see here is a mammalian tree of life, and on the outside, in these different shapes, are the different types of teeth, incisors, canines, or molars. And whenever the symbol is filled, it means that dental calculus is present in this particular study species. And I hope you can see that there are only a few areas in the tree where we didn't find dental calculus, but it's actually present in the vast majority of mammals we have looked at. It does look very, very different to what people would usually consider dental calculus. It can be dark, it can be a film, it can be shiny, it can be, kind of solid deposits. So is this, in fact, dental calculus, or is it just dirt? And that's where Markella comes in. She has analyzed data for about 35 species from which we collected this deposit, and could actually show that a lot of it has a resemblance to oral microbiome. So here, every row is an individual, they're grouped by different species. Whatever is red has a correspondence to an oral microbiome. Whatever is gray is something that we don't know about. What Markella could show is that there is this clade, and these are actually the primates, that look like they have a lot of oral microbiome. But we don't actually think that primates have more oral microbiome in their deposit than the other animals. It's just that we're using the human oral databases to say what is oral and what isn't. So I think what we're seeing here is a reference database bias. So Markella then decided to carry on and to focus on these 35 species for which we have some sequencing data. And it's actually a really nice collection. So she has representatives of almost all major mammalian orders. She has species that differ in their dietary preferences, from herbivores to frugivores, omnivores, and carnivores. She also has 3 independent transitions to the marine lifestyle in her data set. And these are very, very fresh analyses, so we're just scraping the surface of what's possible with this data set. But she was able to show that, yes, indeed, similar to what we heard from Rachel about the gut microbiome, we see a signature of host order and host diet. So I hope you can appreciate these different placements on the ordination for different host orders, but also, for instance, for Artiodactyla, so hoofed animals, the majority of them are here, and they're herbivorous, but the one omnivorous Artiodactylum, which is boars and pigs, they cluster separately, suggesting that there's an effect of diet. Markella then carried out some differential abundance analysis, just really quickly looking at the data, and found some things that made a lot of sense to us. So for instance, among primates, she found 3 bacterial genera, Cardiobacterium and Gamella. 2 of them are commonly present in the human oral microbiome. They are related or associated with heart conditions and heart infections and diseases. One of these genera, Selenomonas, was actually highly abundant specifically in frugivorous primates, and in humans it is associated with obesity, potentially suggesting that the host dietary preferences for more maybe sugar-containing or easy carbohydrate-containing diets can promote the sorry. Yeah.
Change the mic a little bit.
Okay. Thank you. Brilliant. Thanks a lot. So can promote the abundance of these bacteria. So that kind of makes sense, it's something that we would expect. Things that make less sense to us is, for instance, this one. So we find this Actinobacillum to be highly abundant in Artiodactyla, so in these green guys here. The only thing we know about it is that it's mainly found in the urinary tract and kidneys. We do find it in the oral microbiome. We don't know what that means. Right, so this is very, very fresh and, again, very on the surface, and there's a lot more that can be done with that. But I would like to take the rest of the time to tell you 3 microbiome stories. One story is on microbiome and its involvement in what we think is adaptation. A story about microbiomes and temporal sampling and trying to understand environmental contamination, and a half story about hologenomics and population declines. So let's start with oral microbiomes and local adaptation. And for that, we went to one of our favorite study species in the lab, gorillas. And this is the work that Markella has carried out while she was still a master's student, together with a former postdoc in the group, Adrian Forsyth. So quick introduction to gorillas. Gorillas have discontinuous distribution in Central Africa. We have Western lowland gorillas in the west, and as the name suggests, they range at low altitudes. Fruits are quite abundant in their habitat, and whenever fruit is available, they would consume fruit. Across the Congo Basin, so quite a while to the east, we find the eastern gorillas. They're split into closely related subspecies. These subspecies have diverged from each other only 10 to 14,000 years ago, so they're extremely, extremely closely related. And probably one of them is familiar to most of you, the mountain gorillas here. Maybe you've seen gorillas in the mist, you've heard about Dian Fossey, so these are the gorillas that she has made famous. They range, as the name suggests, at high altitudes. Just because of this altitudinal location, fruit is actually hardly ever present in their diet, just it doesn't ripen there. Very, very close to them, just across the Rift Valley, are the Grauas gorillas, so they're sister subspecies. These guys have quite a wide altitudinal range, so they can range as far down as the Western lowland gorillas and almost as high up as the mountain gorillas. And depending on their habitat, they have varied availability of fruit in their diet. So great. So Markella and Adrian decided to see if there are differences in the oral microbiomes of these 3 relatively closely related gorilla taxa. And indeed, they found differences, but not in the way we would have expected. They found that the mountain gorillas are, in terms of the composition of the oral microbiome and the function of the oral microbiome, significantly different from both Western lowland gorillas, and they're very, very close, phylogenetically close, and geographically close sister subspecies, the Grauas gorillas. We couldn't explain it for a while until we considered altitude. And it turned out that indeed altitude is the only factor that kind of helps us make sense of this data. And this is because since Grauas gorillas range at different altitudes, we could subdivide this data set into low altitude Grauas that were absolutely indistinguishable from Western lowland, and high altitude Grauas that had higher similarity to mountain gorillas. So we believe what is happening here is kind of diet-mediated microbiome change that is tracking the altitudinal differences. What Jala has presented as well, we can actually go beyond simply using reads and metagenomics. We can try to assemble metagenome assembled genomes, and that's what we did for this data set. And one of the metagenome assembled genomes that we could put together with extremely short reads, so those people who were saying, oh, it's really hard with long reads, we're working with reads that are about 60 base pairs long, but it still works. So one of the genomes is Neisseria, a typical oral bacterium. Our MAG shown here was clustering close to human isolates of the same genus, which makes a lot of sense. But what is interesting about Neisseria is that it is involved in nitrogen metabolism. And I knew nothing about nitrogen metabolism, to be quite honest, when we started this project, but it turns out it's actually quite interesting. So when we consume leafy vegetables or we drink beetroot juice, as shown here, we are taking in a lot of nitrate. But we're actually not really good in metabolizing nitrate. So what is happening is that it goes into the stomach, from the stomach it goes into the intestine. We can't do much about it. It's being taken up by blood and it's being enriched in the saliva. And saliva presents nitrate to oral bacteria that can metabolize it and turn it into nitrite. And one of these oral bacteria is Neisseria. Nitrite, that we can deal with, so we swallow it back in, and then we reduce it to nitric oxide, which is a physiologically relevant molecule which is associated with oral health and cardiovascular health. But it's also increasing sport performance, and it is increasing physiology at high altitudes. Did I just say altitudes? I think I did. Didn't we say there was something about the gorillas and altitude? So we were blown away by that, and we're like, okay, let's see, do we have higher abundance of these bacteria in high-altitude gorillas? And indeed, it turns out yes. So we found a high abundance of Neisseria and also some other nitrogen-reducing bacteria in mountain gorillas compared to the other two subspecies. We also, because we have MAGs, we can actually look at the presence of the relevant genes, and we could show that our MAG here has the majority of genes that are involved in nitrogen metabolism, and actually the only two here that are absent are not absolutely essential for nitrogen metabolism. So although we don't have direct evidence, we have some suggestion that this bacterium and its presence in the oral microbiome may contribute to the high altitude habitat or adaptation or ability to live at these high altitudes for mountain gorillas. And this is very exciting for us because my group has been looking at mountain gorilla genomes for years. And obviously, you know, them living at high altitude is always a question that comes up. Can we find anything that explains how they persist at high altitudes? And we couldn't. Nothing convincing. So here, for the first time, we have this link between high altitude adaptation and a humble oral bacterium. So with this, I would like to move into this conceptual figure again and tell you the two remaining stories, one of them on this part of the story about external environments and climate change and pollution. And in particular, I would like to focus on the environmental contamination with human-made products, and specifically antibiotics. We all know antibiotics are extremely important. They saved millions of lives, but there is a dark side to the antibiotics, and this is antimicrobial resistance. So here, a quick sketch about the antimicrobial resistance cycle. So we as humans started mass-producing antibiotics in around 1940s, and the antibiotics are being predominantly used in two sectors, the biomedical sector over here and the agricultural sector. And of course, humans and animals, we interact with each other, we eat, we care for them, we spend time together, so we exchange trace antibiotics and resistant bacteria during these interactions. They also get out into the environment through sewage, farm runoffs, and things like that. From the environment and through these various interactions, they're being picked up by wild animals. Of course, wild animals and domestic animals interact, wild animals and humans interact. So all of this promotes the exchange of antimicrobial resistance, be it as trace antibiotics or resistant bacteria, and this connection or this exchange between humans, livestock, wildlife, and the environment is very nicely encapsulated in the One Health concept. So if we want to understand how human activities influence antimicrobial resistance in the environment and the wild animals, we could, of course, just go out and try to quantify it. But there is a problem. And the problem is that antimicrobial resistance is actually a natural part of the natural environments. This is where we got our antibiotics from in the first place. So going somewhere and quantifying AMR is not going to tell you how much humans have influenced it. So instead, I'm arguing we need to take the temporal approach. So we need to go back to the time before humans started mass-producing antibiotics. And this is exactly what Jella, who spoke before me, did in the lab when she was still good enough to work with me. Anyway, so Jella decided to do the most logical step you can possibly imagine and study Swedish brown bears, because it makes total sense to try to answer this question in Swedish brown bears. And I will try to convince you that, yes, indeed, it did make sense. So number one, brown bears are widely distributed across Sweden. So everything that's colored here, this is where brown bears occur today. In ancient times, they also used to occur in the south. They're omnivores, so they have diverse diets. They explore different habitats, but they do try to stay away from humans as far as possible. So they're indeed really wild animals. A really kind of pragmatic reason is that the Natural History Museum in Stockholm has an amazing collection of brown bear skulls that dates back to about 180 years, so well before mass production of antibiotics started. So what Jella did was take these museum collections and quantify the amount of antimicrobial resistance in brown bear individuals by looking at their preserved oral microbiomes. So every single dot here is a brown bear dental calculus sample from a single individual, and we quantify antimicrobial load, so the abundance of resistance genes through time. So this is our naive time period, because before the 1950s, no mass production of antibiotics has been carried out in Sweden. And then over the next 5 decades or so, we see an increase in antimicrobial resistance, which also tracks down the increase in the use of antibiotics in Sweden. What is interesting about Sweden is that it was one of the first European countries to actually recognize the danger of antimicrobial resistance and to introduce control measures for that. So in mid-1980s, antibiotics were banned from animal feed, and in mid-1990s, there was a very stringent governmental program about the use of antibiotics in the biomedical sector. So what is interesting that if we look at bears that spent their entire lives after these two measures were introduced, we actually see a reduced amount of antimicrobial resistance in them. It's not on the same level as our naive samples, but it's still a significant reduction from the previous decades. This is a different picture than if we look at the diversity of antimicrobial resistance genes, which are actually increasing through time and are tracking down the diversity of antimicrobial classes that are being used as drugs. So I will finally, for the last 5 minutes, so maybe 3, I'll tell you a very, very short half story about our first attempt to look at this particular factor. So how population declines affect the host populations and interact with microbiomes. And the logic behind that is following. As populations decline, so does their genetic diversity. So this is something that Tom has shown during his PhD with gorillas, that Tom here has shown multiple times in many other species. So as genetic diversity declines, so may the diversity of immune factors and immune genes. This may reduce the host control over microbial communities and lead to proliferation of specific organisms that may cause diseases or reduce host fitness in some way. So this is the framework we're using to operate on. And for that, we're going to stick with the brown bears in Sweden, and this is because they have been actually hunted nearly to extinction in 1930s, with populations numbering only about 130 individuals. A hunting ban was then implemented about 15 years after that, and the population has recovered to about 3,000 individuals today. But this bottleneck has indeed caused reduction in genetic diversity, so the populations that live today have considerably reduced genetic diversity compared to populations before the bottleneck. So while we were working on Jella's project, we also realized that brown bears actually have a lot of oral diseases, and that's not something that I would have expected from a wild animal. So I hope you can see how inflammation has eaten away the bone here, how we have these cavities in the teeth. So we became interested in that, and 2 master students in the group, Maya and Zhang, have spent hours and hours in the Swedish Natural History Museum scoring brown bear skulls for presence and abundance of oral diseases. And then Adrian took on this data and plotted it against population sizes. So what you see on this graph in black are the population sizes. So this is the bottleneck. We don't really have a lot of information about population sizes before that. And then we have the steep recovery. And in blue, you see the prevalence or abundance of oral diseases in brown bear skulls through time, with prevalence reaching almost 50% of specimens being affected during the times when the population is at its lowest. So what we see is this kind of reverse relationship between disease abundance and population size, which indicates that there may be some interactions between host genetic diversity or host density and oral diseases. So Jella, in one of her previous works, actually analyzed a diseased sample. It was this sample in particular, and she could show that in the sample with caries, we have a really high prevalence of bacteria that are known to be caries-causing in humans, and these bacteria are virtually absent in healthy bears. These caries-associated bacteria are involved in sugar fermentation pathways, in acidification pathways, so it makes a lot of sense. Adrian then took on this data and expanded the data set to show that diseased bears indeed have very distinct microbial communities compared to healthy bears, that their microbial communities are reduced in diversity compared to healthy bears. So this is where we stand now. The clear, obvious thing that we're missing is host genomic data, so that we can establish this link between host immune factors or genetic factors and the microbiome, and that's what we're hoping to generate next. So to sum up what I have been talking about, I hope I could get you a little bit enthusiastic about the diversity of the preserved oral microbiomes from ecologically and phylogenetically diverse species. So if you think of your favorite species, if it has teeth, chances are it has dental calculus, so chances are you can try to come up with questions or study questions that would involve the temporal perspective. I also believe that these studies of the microbiome through time actually open doors to a lot of things that we never considered in the past. I've given you a taster for environmental effects. I've given you a little bit of a thought about the demographic processes of the host. But you can think about anything you consider. You can think about hybridizations and how this may affect the interactions of the host and the microbiome. You can think about ecological effects such as range expansions, invasions. All of this can be studied from the temporal perspective by taking the hologenomics approach. And I would also finally like to highlight the tremendous value of natural history collections, because I think they're sometimes underused and definitely underloved. And with this, thank you so much for your attention. Oh yeah, and sorry, this, the most important one, of course, tremendous thanks to all the people who ever agreed to work with me over the years, to our collaborators, and to natural history museums for supporting us. Thank you now.
Thanks for a great talk. I believe we have time for one or two few questions. Yeah, Tom has a question.
Thank you, Katja. It's very lovely. I love the temporal hologenomics. With regards to the gorillas, do you think there's a genetic factor in the gorillas also explaining the microbiome difference you're seeing, or is it purely just diet driving that relationship?
We have no idea, and obviously for that we would need to link gorilla genomes and the microbiomes, ideally from the same individuals. We have not looked at that. I do not think we have the data set, so basically the number of individuals to look at that yet, but it should be possible in the future.
So we just need to hunt more gorillas?
Oh no, we need to study more gorillas from the museum specimens, because indeed there are hundreds, if not thousands, killed gorillas in the museums. So definitely not hunting.
Anton?
You showed species with different dietary habits, and do you know whether there are any biases in how this classification will be captured in the microbial community from the oral cavity, and whether these biases could be driving the pictures that you see? I mean, how to tease apart what was originally in the mouth and how this got captured in the
Oral microbiome, yes.
classification of the calculus?
Yes. So it's a very valid question, because what we are looking at is indeed not the oral microbiome, but the dental calculus microbiome. So whatever manages to be preserved in the dental calculus. And there indeed could be differences between what's captured, but on the other hand, it is a living biofilm. So whatever this biofilm is made up of, this is what's going to calcify over time. I do want to highlight one interesting aspect, since you touched on diets. It's actually, diet is a big determinant of whether or not you're going to have dental calculus built up, yes or no. And in particular, it is the proportion of proteins and fats in the diet that will determine whether dental calculus is going to build up. Hypercarnivores do not have dental calculus, or rarely build up dental calculus. But carnivores that have at least some proportion of carbohydrates in their diet do. So that's in terms of thinking about whether your species has it.
Okay. Thank
Thanks
you.
so
and
much.
additional questions will have to be
Absolutely.
covered during
Thanks.
lunch. Thank you. And let's move on.